Hello, this is Dr. Kyung-Soo Ham from BioLeaders Corporation. I'm very happy to introduce our company. BioLeaders Corporation was established in the year 2000 by 20 bioscientists around the country in Korea. The company is currently a public company. It was listed in Korean stock market called KOSDAQ in 2016. Currently, we have around 67 employees. Half of them are involved in R&D. Our key personnel is our chairman is Andy Park, who was the former president of, former CEO of global business of Daewoo Group company, and we have vice chairman called Chang-Gyun Kim. He's the former CEO of Sinpung Pharmaceutical Corporation. We have CEO Dr. Sang-Seok Kim, who was the former CEO of General Management at Celltrion Pharmaceutical Company. We have advisor Dr. Moon Hee-sung. He's the creator of this company. Currently, he's working as a professor in Kookmin University. We have Director of New Drug Development, Dr. Kwang-il Chung, who was the former senior scientist at Pfizer and Sanofi, and he also worked at LG Life Sciences. Myself, I'm in charge of the global business development. We have two proprietary platform technologies. One is called the HumaMAX Technology, and the other one is called the Mucomex Technology. We have a couple of items from HumaMAX Technology and two drug candidates using Mucomex technologies. The first proprietary platform technology is HumaMAX Technology. This is basically polygamma glutamic acids utilizing technology. Polygamma glutamic acid we isolated and we discovered from Korean traditional food, and it shows that it can stimulate various immune responses. It can induce innate immune responses, also adaptive immunity. It can also inhibit myeloid-derived suppressor cells, so it can increase tumor-infiltrating lymphocytes. Since the material was isolated from Korean traditional bean-based food, so this is very safe. This is glutamic acid linked with gamma linkage. It's an amino acid biopolymer. It's a high molecular weight, around 2 million Daltons. It's biodegradable, water soluble, as also anionic, and it's very safe. The experimental data shows that polygamma glutamic acid has very strong immune stimulating activities, such as it can activate macrophages, it can activate dendritic cells. It can also increase interferon gamma levels from spleen samples, and also it can activate natural killer cell cytotoxicity. Polygamma glutamic acid binds to toll-like receptor 4 of the immune cells, and it can go through both MID88 independent and dependent pathways to induce cytokines and also to stimulate natural killer cell cytotoxicity. We have patents of polygamma glutamic acid, and also we have patents of our master cell bank, and we have various usage patents. It's about BLS-H01 developed using HumaMAX technology. When a woman gets infected with the Human Papilloma Virus, the disease progresses from SIN1, SIN2, SIN3, and finally cervical cancer. Up to SIN3 stage, there are no drugs available. And when you reach SIN3, the hospital recommends you to do the little surgery called colonization, and has a very strong side effects such as infertility, abortion, premature birth, and so on. So BLS-H01 is first-in-class oral drug targeting SIN1 stage. We did human clinical trials. Phase 2B clinical trial was completed. And the result shows that the cure ratio of using a polygamma glutamic acid was higher compared to the placebo. The difference was like 15%. It can also shorten therapeutic period from 6 months to 3 months. Since BLS-HO1 is first-in-class oral drug, there is no market established, and we can only estimate using the number of patients. Currently, the whole world, about 6.6 million patients are in SIN1 condition. We can also estimate the market size by looking at the HPV vaccine markets. Since the BLS-HO1 targets a very early stage of human papilloma virus infection, we asked the physicians whether we developed the drug. They are willing to prescribe the drug, and indeed most of the physicians says they will prescribe the drug. Next one is BLS-HO3. This is polygamma glutamic acid with chitosan. It's a nanoparticle adjuvant. The experimental data shows our nanoparticle adjuvant can induce 2 to 10 times higher antibody responses compared to the commercially available adjuvant, MF59 or alum. It can also induce 5 to 10 times higher T-cell responses. In addition, after vaccination, the antigen-specific immunoglobulin titer can be maintained up to 24 weeks. Since polygamma glutamic acid has very strong immune-stimulating activities, such as it can inhibit myeloid-derived suppressors, it can also increase CD8-positive tumor-infiltrating lymphocytes, it can activate T cells, and it can also activate antigen-presenting cells. So we think the polygamma glutamic acid can be worked as a combinatorial therapeutic, such as anti-PD-1 monoclonal antibody therapeutics. Indeed, the data shows that polygamma glutamic acid exhibits a synergistic anti-tumor effect in combination with the anti-PD-1 monoclonal antibodies. Second platform technology is called the Mucomex Technology. The technology is basically antigen display technology on the surface of lactic acid bacteria. As you know, lactic acid bacteria is a gross microorganism, so the technology is very safe. The final product will be enteric coated, so it's an oral drug we are developing. So Mucomex technology, we have a lactic acid bacteria, and then we are using PGSA as a display motif, and then we can put any type of antigen on top. We have patents individually or whole system. The mode of action of this Mucomex technology is when you take it orally, it goes through, pass through stomach and reaches to Peyer's patch to reach to the effector sites to induce mucosal immunity. We have patents. BLS-MO7 also registered as a HumaMAX. It's targeting SIN2 and 3. As I said, there are no currently available drugs, so this is first-in-class drug. We are using E7 antigen of human papilloma virus 16 as a target antigen. So the drug candidates, when you take it orally, it can induce E7 specific cellular immunity, and also it can induce E7 specific immunoglobulins. Investigator-initiated clinical trial was done in University of Tokyo, and from the result, we use 1,000 milligram per day was the optimum dosage. Using that, we did phase 1-2A clinical trials in Korea. And the result shows out of 8 patients with SIN3, which is severe dysplasia, and out of 8 patients, 3 became normal and the other 3 became SIN1. So the overall response rate was 75%. Currently, we are doing phase 2B clinical trials in Korea. There are around 16 hospitals are involved, and the clinical trial is almost completed, and the final report will come out this coming late October. Again, this is first-in-class drug, so we can only estimate by looking at the vaccine markets. The last one is BLS M22, which is targeting Duchenne Muscular Dystrophy. As you know, Duchenne Muscular Dystrophy is a rare disease. The symptom onset is in early childhood between ages 2 and 3, and most of the DMD patients do not survive beyond their teen years. Others trying to develop therapeutics for Duchenne muscular dystrophy using gene therapeutics such as stop codon read-through or exon skipping. Some companies are trying to develop anti-myostatin monoclonal therapeutics. However, our BLS M22 can induce anti-myostatin antibody by active immunization. The table shows our competitors. Some experimental research using MDX mice shows that indeed BLS M22 induces the serum anti-myostatin antibody levels, and as the antibody level goes up, the body weight also improves, and at the same time, movement also improves. We obtained Orphan drug designation from US FDA in 2017, and also OTD was granted in 2019 from Korean authorities. Currently, we are doing phase 1 clinical trials. The phase 1 will be completed by the end of this coming October. Once we finish the phase 1 clinical trials, we plan to do the phase 2A clinical trials targeting, in addition to DMD, also we think it can work with ALS, SMA, or sarcopenia. So phase 1 for the M22, phase 1 is ongoing, and we are currently doing efficacy tests in different disease conditions or muscle wasting conditions. So from HumaMAX Technology, PLS-H01, as I said, we completed the phase 2B clinical trials. Currently, we are actively looking for a partner to do the global phase 3 clinical trials. From Mucomex Technology, PLS-M07, currently we are doing phase 2B clinical trials. Again, I'm looking for a partner to do the global clinical trials or license out. PLS-M22, which is early stage. However, I'm still looking for a partner to do the global clinical trials and also license out. Thank you.